Now that we've gone through the structure of DNA, the actual molecular structure, let's back up a little bit and talk about how that information that we find encoded in our DNA is actually expressed or used in a living organism. So let's talk about the actual unit that is used for a specific characteristic. So that unit is the gene. So the gene was talked about well before the structure of DNA was actually deduced. It came into common use in the early 20th century. And now defining what a gene is a little bit more challenging because it sort of depends on the situation in which you are in. Genes have many different structures and they have many different roles within an organism. But in general, a gene was, especially before we had the structure of DNA and sequence knowledge that we have now, was sort of the smallest particle that was related to a hereditary characteristic. So this term pangen was coined to sort of identify some unknown factor that was being passed on from parent to offspring. In general, a gene is a short segment of DNA that codes for a specific protein and therefore a specific characteristic. We're going to define gene many different times along the way. We're going to sort of refine it as we get farther along in our discussion of genetics. But keep in mind that when we're talking about a particular characteristic, we are talking about the genetic component or the small segment of DNA that actually functions for that to produce that protein. Defining a gene is a little bit hard, but all genes have specific characteristics. Two important characteristics are replication and expression. So a gene has to be replicated before it can be used in expression or before it can be transmitted to the next generation. Now, this is important because if you're going to pass on your genetic information to either the next round of offspring or just one of your cells passing on genetic information to the next generation of cells, you want to keep a copy for the previous cell or previous generation. So that piece of DNA or the whole piece of DNA needs to be copied before it can be passed on. Really important factor is that during the replication process, there is a potential for introducing errors or variation. Now, this variation is really important in evolution. So as we replicate our DNA and pass it on, it can be changed. So that means that DNA is not static. It can change over time. If a piece of DNA is going to be useful in an organism, it has to be expressed. So you have to have a way to get the information for the gene that your cell needs out of the larger piece of DNA in order to use it to produce a phenotype. Now remember that a phenotype is the physical characteristic or the characteristic of an organism that you're looking at. So maybe it's a physical characteristic, maybe it is a enzyme and a metabolic pathway, something like that. So all of these characteristics relate to the structure of DNA. Remember we said that DNA has two complementary strands. That complementarity is really important if you're going to copy your DNA. You have to have a template from which to copy. And then if you're going to express it, you need to have a way to extract that information. And you extract that information using the complementary nature of DNA. So this whole process of taking your information from your genetic code and turning it into a phenotype is called gene expression. Now, the process of gene expression is described in something that we call the central dogma of molecular biology. Now, this was a term that was coined by Francis Crick in 1958, and it shows or describes how we actually get our information from our genes in our DNA all the way out to our phenotype. So during this process of gene expression, you take some information from a specific gene within your DNA. Now, the first thing that you're going to do is you're going to transcribe it. That means you are going to copy it into the same language. So if we think of DNA as a manual, a sort of a owner's manual. So if DNA is our manual, and we want to use this information for something, we want to extract just the small bit that we need right now. So we might want, let's say, just a chapter of our manual. The gene is going to be comparable to a chapter in our manual. So let's say that you want to change the oil in your car. You don't read the whole manual every time. You go to the section that explains the oil change and what type of oil you need and what steps you need to do it. That is the information that we would be pulling out from our gene, just the little bit that we need to make the particular protein that we're interested in. So you might jot down some notes. That would be equivalent to transcribing your genetic information. So if we take the information in DNA and we take out just the little bit of information that we need, we do this through a process called transcription. And in that process, we make RNA, which is also a nucleic acid. So we're keeping the same language here. It's just a slightly different dialect. There's some differences between DNA and RNA, but it is a copy. RNA is a copy of the gene we need. And then in the second step of our gene expression, we are going to translate that information from RNA into proteins. Now, proteins are written in a language called amino acids. So we are actually translating from nucleic acid to amino acid. So this process of genetic expression or direction of information goes from DNA, or the information encoded in nucleic acids, all the way to protein, or the information that is actually expressed in the cell. So here's a different diagram to sort of show the same thing. If we look at our template DNA here, the DNA that we're interested in using, we're going to make a single-stranded copy in the form of RNA. And then RNA is actually going to be used as a template to make proteins. Now, this RNA template is a 3-letter code, and every 3 letters is going to code for an amino acid. So we are going to use that RNA template to string together a chain of amino acids. A chain of amino acids is called a polypeptide. So we're going to make a polypeptide, and that polypeptide is going to eventually be processed and turned into a protein. That protein is what is going to produce our phenotype. So something like eye color, shapes of the pea pod in a pea plant, antibiotic resistance, blood type, things like that. But after Francis Crick coined this phrase, central dogma, a lot of research continued on, and we actually have sort of an expanded view of the central dogma at this point. And this includes DNA replication. So this is where we are going to take the information in DNA and copy it into another molecule. And this is our DNA replication. This is going to be what is transmitted or passed on. And then, of course, we have our RNA molecule that is going to be turned into a polypeptide. Now, we can also have some other types of molecules that are made up from our DNA molecule. And these are called non-coding RNAs. We'll talk about those later. They perform different jobs within the cells. But we're mostly going to focus on this idea that all of our genes are going to produce a polypeptide and eventually a protein of some sort that makes a phenotype in an individual. So we talked about the structure of DNA, but RNA is really important in this gene expression process. And it differs in a few ways from DNA. In this molecule on the left, we have our DNA, and you'll recognize the ribose sugar, the phosphate groups, and the nitrogenous base, and then, of course, that deoxyribose, that 2' carbon missing the oxygen. RNA is a slight modification. RNA is a ribose sugar, but that oxygen is present. So RNA has that hydroxyl group attached to the ribose sugar. Another major difference is that DNA contains the base thymine, but RNA contains the base uracil. Now, this relates to the fact that RNA is sort of a temporary molecule. Uracil is cheaper to make, and it degrades a lot faster. And so it's okay that RNA is made with uracil. But DNA needs to be more stable, so it needs that more stable thymine molecule. So RNA is very, very similar to DNA, with the major exception of the hydroxyl group and the uracil. RNA is ribose nucleic acid, so very similar to DNA. Uracil and thymine only differ a little bit. So thymine and uracil really just differ in this functional group attached to one side of the ring. Like uracil and cytosine, like thymine and cytosine, uracil is a pyrimidine. And it is basically the counterpart for thymine in RNA. So because uracil is a little bit less stable, you can incorporate uracil into RNA, which is also cheaper for the cell to make. And you can actually make it by modifying cytosine if you have to. All right, so during the process of transcription, which is the process of going from DNA to RNA, we are going to use the one strand of the DNA molecule. This is our gene. And we're going to take only one side of it, so a single strand, which means we have to open it up and have access. Now, these strands are called the coding strand. This is where your DNA gene is located. So this is the actual sequence of the gene. Now remember that these are complementary. So if we open them up and use the template strand to make a complement, we actually get our coding strand. So this was the coding sequence now in RNA. That is the same as the coding strand in our DNA. So now we have, by using the complement, making the complement of the template strand, we have the same strand as our coded gene. We cannot use the coding strand because otherwise, if we do that, we get the complement, and that's not going to produce the protein that we want. So when we use the coding strand of our RNA, we are going to take that RNA strand and use that to make our protein. So our DNA coding strand is going to be the same as our RNA that is used to make the protein. Now that RNA is called messenger RNA, because it is taking the genetic message from DNA and using it to make a protein. All right, so when we look at the overall makeup of chromosomal DNA, the overall strand of DNA, we have a lot of different places where we can sort of hide genes. Now, remember that a strand of DNA is double-stranded, or I'm sorry, a molecule of DNA is double-stranded. And we can have genes encoded on either side. So we can have genes on the top strand, or we can have genes on the bottom strand. The other thing that's cool about DNA, particularly bacterial DNA, where you have a small amount of genetic information, is your genes can actually overlap. So you might have a gene occurring on one side running this direction, and then a gene occurring on the other side running in the opposite direction. And so you can have overlapping regions that allow you to utilize the limited amount of space in a particular genome. So both sides of the DNA can be used as a template. So we can use either side to encode our genetic information.